Now protein, structure of protein. They are heteropolymers. They are made up of long chains of amino acids. Amino acids, they form a long chain polypeptide. So now proteins, they are made up of amino acids, long chains of amino acids. Further, they construct themselves in such a manner to perform certain functions. Let's see what construction they do to become complex and perform different functions. Now, in inorganic chemistry, you would give just for NaCl only a formula, NaCl MgCl2. But in the organic chemistry, there is two-dimensional structure for benzene, naphthalene, and the physicist would conjure, they'll construct the structure to be three-dimensional, but the biologist, they will construct to be four-dimensional. Let's see how. Now, this is the chain of the amino acid which will become protein. They will construct the position from where it starts and where it ends. Now left end or where it starts is known as N-terminal, and where it finishes it is C-terminal. So when it is a long chain, it is only one chain, then it is primary. It's the primary structure of protein. Now, when this primary will twist, it will become helical. So that is secondary structures. Here you can see the helical like this. It forms the secondary structure. Here this is the cartoon of the secondary structure. You can see how the helix has formed, curling around itself. Now full protein won't be there as secondary. Only a part right hand side there will be a secondary structure. So this is how it will lead. This primary, a long chain will form helix. This is alpha helix. This is helix. Now this is beta plated sheets. They will form sheets. The amino acids will arrange themselves in sheets with the peptide bonds, attached with the peptide bonds. So this alpha helix and this beta plated sheets, they are arranged in the secondary structures. Primary will be long chain, and this is the next step. They will construct into helical structure like this or like this. You can imagine, because more complex it will slowly become. So this polypeptide chain arranged into helix, that is the alpha helix, and here it is arranged in the beta plated sheets connected with the peptide bonds. So this is the secondary structure. So from primary, now they have formed secondary structure, but they will form tertiary structures like a hollow woolen ball. How the woolen ball is formed? The threads are arranged like this, like this, like this, then like this, like this, like this, like this, like this. On all the dimensions the threads are arranged. So these threads are of polypeptide will be arranging like a hollow woolen ball. So that will be tertiary. You can see they are connected by the hydrogen bond, the disulfide bonds. So from here, primary to secondary, now they have become more complex, tertiary. But now they have the capability of developing more architecture, making it more complex. Different types of protein, they entangle with each other to form complex protein to perform complex functions. So here they form quaternary. two types of protein, they will entangle with each other to form a complex quaternary protein. Now, hemoglobin, human hemoglobin, they are formed from the alpha and beta. Two types of alpha and two types of beta, four types, they entangle with each other to form the quaternary structure. So they are able to perform a complex functions. So this is how. Now this is the cartoon structure of secondary forming helix, and here you can see the tertiary structures forming like a woolen ball which is entangled this way, this way, this way. There are many threads which are there, which are entangled, which are formed in a structure to have to form a tertiary structure. So here this is how the proteins, different types of proteins, see, we all are formed by the proteins. They perform complex function, they have complex structure. Why? Because they are formed from this polypeptide, one chain of amino acid. They form helical structure. This is example here, you can see alpha helix. They form plates, beta plated sheets. Then they form tertiary structures like this, and they form a complex tertiary structure. Then they form quaternary structure where different types of proteins are entangled to form a complex protein. Now, nature of bond linking monomers into polymers. Monomers is one molecule, they form long chain to become polymers. Now, amino acids, they are one molecule, they are bonded together with the peptide chain to form a polymer. Now, in the amino acid, the carboxylic group of one molecule bonds with the amino group of the other amino acid. Carboxylic group and the amino group, they bond together, and in the process the water comes out. Water moiety, water part comes out while forming the peptide bond. So, a peptide bond is formed between the two amino acids. Then they get chain, they form chain. They just go on linking. Carboxylic group of one amino acid getting attached to the amino group of the other amino acids, and they form long polypeptide chain. They are linked by a peptide bond and they form a long chain. So from one monomer they become a polymer. Now, glucose, they form glycogen, they form polymer. From monosaccharide they become polysaccharide. So one glucose gets attached to another glucose and form the polymer. So in the process, the carbon and carbon of glucose, they bond together and water molecule comes out. So here also dehydration takes place. Water moiety, water part that comes out, and that leads to the formation of glycosidic bond in glucose. This is how monosaccharide, they become polysaccharide by glycosidic bonds. Now the nucleic acid. Nucleic acid, it is made up of sugar, it is made up of phosphate group, sugar. There's a bonding which is present in between the sugar. The third carbon, fifth carbon, they get attached, they form the bond. You can see this fifth Third, there's a bonding which is formed. Here, the phosphate group gets attached to the hydroxyl group of the sugar, and that is the ester bond is formed. Here, two ester bonds are formed, therefore it is known as phosphodiester. Two bonds are there, two ester bonds are formed. So here it is the phosphate group, here it is sugar, and here sugar, here phosphate group. There's a long chain which is formed. Attached to the sugar, perpendicular, is the nitrogenous base, and here nitrogenous base attached to the sugar, here there will be nitrogenous base. So thymine, guanine, adenine, cytosine. Here it will be in one series. Now here this DNA has got two strands. They form secondary structures. So according to this Watson-Crick model, that is the B type of DNA, the secondary structures are formed in this way. The thymine forms a bond with the adenine. Hydrogen bonds, two hydrogen bonds are present between thymine and adenine. Three hydrogen bonds are present between guanine and cytosine. Here the other strand will have, this is the nitrogenous base which is attached to the sugar, which is attached to the phosphate group. This is sugar, nitrogenous base. Here also long chain of nucleotide will be formed. It will just go on, thymine, adenine, guanine, cytosine. So here again it will be adenine, thymine this side. They are anti-parallel. They are exactly opposite because opposite nitrogenous bases are present. Here the backbone remains same with the sugar and phosphate group, which has got two ester bonds, diester bonds are formed. And this nitrogenous base, they go on getting attached with the hydrogen bond, and this are opposite. Nitrogenous base are opposite, therefore they are anti-parallel. Now they will form helical structure. Helical structure will be formed like this. Helical structure you can see. Now this two strands, they form the helical structure, which are exactly opposite, therefore they are anti-parallel. Now 10 chains of nucleotide will be arranged. They are 10 nucleotides. They form a chain, a polymer of 10 nucleotides. One nucleotide is formed of the sugar, phosphate group and a nitrogenous base. So this is one nucleotide. So here one strand will have 10, the other will have 10. So they will form helical structure. They are anti-parallel and they twist with each other. And they are bonded by the hydrogenous bond which is formed between the nitrogenous base. Here two bonds between thymine, adenine, three bonds between guanine and cytosine. Now the whole pitch is 34 angstrom and one nucleotide pair will be 3.4 angstrom. So this is the structure which is formed by the bonding. The monomers, they become polymers by the bonds which are formed. So the bonds, for examples, are glycosidic bonds and the diester bonds, peptide bonds. They link for monomers to form the polymers. Now the dynamic state of the body constituent. Dynamic means ever changing, never the same. The change takes place. Now let us see all the organism, protozoan, plant, animal. They have compounds in them, certain compounds which are biomolecules. They are indefinite amount. molar per liter, molar per cells, they are in definite amount. Those are the biomolecules. Now you will see that the biomolecules, there's a turnover, they change, it's ever changing. One molecule leads to the formation of another molecule, another to another, and they are ever changing, and they are changing in a definite path. It's like the city traffic, how the traffic goes on smoothly in a definite path. And remember, here no mishap takes place. The changes takes place very smoothly. Now, this process of forming new molecules one after the other, it is known as metabolism. The biomolecule which is formed is metabolites. There are thousands of chain reaction, that reactions which are taking place, which leads to new and new metabolites being formed. The process is known as metabolism. Now, few examples: removal of carbon dioxide from amino acid leads to formation of amine. Removal of amino group from the nucleotide base is a metabolic reaction. Hydrolysis of glycosidic bond in a disaccharide is a metabolic reaction. So the metabolism takes place, which leads to the formation of metabolites. That is why it is the dynamic state of the body constituent. The body constituents are the metabolites which are formed by the changes, by the chemical reaction that is taking place continuously, and it follows a definite path, and they are interlinked. So there is a junction where the reaction is same, they meet at a junction and then change over to another pathway. So the path is linear and circular, which forms the junction. There is continuous chemical reaction taking place. There is a metabolic pathway which is linear and circular. And new molecules are formed in a definite way. New metabolites are formed. It completes the reaction and goes on. It is never at equilibrium. It is never at, say, a peace. It is ever changing. Therefore, it is dynamic, new, and it comes to the same chemical reaction. So, the metabolism is formation of new compounds, metabolites, which are biomolecules, which are changing from one form to another. So this is metabolism. But in a living organism, what leads to the chemical reaction? Who leads the pathway? Who changes from one molecule to another? Now, in a living organism, the reaction is catalyzed. Nothing takes place on its own. It is catalyzed by the proteins, proteolytic proteins, which are enzymes. So in nature, now carbon dioxide can dissolve in water by itself. It's a physical reaction, but in living organisms, the same dissolving of carbon dioxide in water is catalyzed. It's a chemical reaction. It's a metabolic reaction. So it is catalyzed. Now, in living organism, continuous reactions are taking place in living organism. So, which is the hero which is leading to this chemical reaction, continuous chemical reaction? It is the enzymes. Enzymes are proteins. They are proteolytic. They make these biomolecules change from one form to another. And this is how there is a dynamic of metabolism in the living organism. It is ever changing. Now, metabolic basis for living. When you are living, metabolism takes place. Metabolism is of two types: anabolism, catabolism. When construction takes place, anabolism; when destruction takes place, catabolism. Both are very important for our living. So, what happens in anabolism? Now, the metabolic pathways, they convert the complex structure from simpler one. In the metabolic pathway, complex structures are formed from the simple one. For example, from acetic acid, cholesterol is formed. So here construction takes place, that is anabolism. When you construct something, energy is required. Energy is required for construction. When destruction takes place, then the energy is given out. So here, in the case of conversion of glucose, that is glycolysis, which takes place in the 10 steps, which leads to the formation of lactic acid, here energy is released. We get energy by this process, which is in the form of chemical bond, that is ATP, adenosine triphosphate. It is stored in our body whenever it is required for work, converting into work. We get this energy from ATP, which is adenosine triphosphate, which is the bond energy, which is the chemical bond energy, which is formed due to glycolysis, the process of glycolysis, conversion of glucose into lactic acid, which is a catabolic process. Now there are many metabolic processes which are happening. Now amino acid getting converted into proteins, that is construction. You are building something, so energy is required. That is anabolism. Construction takes place. Assembly of amino acid requires energy. Now, catabolic pathway leads to the release of energy. Whenever breaking takes place or catabolism takes place, energy is released. Classic example, glycolysis, which is a chemical bond which requires energy for various biological processes like biosynthetic, osmotic, pressure, mechanical work. All this work is done with the help of energy. Energy is in the form of ATP, adenosine triphosphate. So all living organisms have a metabolic process of making energy, storing energy, and using that stored energy to do work that forms bioenergetics. Now there are thousands of compounds which are formed in our body. Those are molecules, those are metabolites, but they will be in definite amount. For example, the blood concentration of glucose, it has to be from 4.2 millimoles per liter to 6.1 millimoles per liter. It cannot be less or it cannot be more. If it is like that, then it is unhealthy for our system. So for a healthy system, the amount of biomolecules, the organic compounds, has to be definite. Now if you see hormones also have chemical changes in our body. They have to be definite. They are measured in nanograms per milliliter. Now the biomolecules which are formed due to metabolism. So that definite number, the number of biomolecules in a cell, is known as metabolic flux. So steady state has to be maintained in the cell for the metabolic path to follow smoothly. So the amount which is present of each biomolecules, each compound, that is the metabolic flux, the path which is followed due to this metabolic flux. Now a steady state has to be maintained. Now here in a living cell, the steady state is not equilibrium, because Equilibrium means the reactions will stop. It will be a steady state which does not lead to any reaction. So in a living cell, the steady state is non-equilibrium. Now, non-equilibrium is able to perform work. It is able to perform work. Why? Because there is input of energy due to metabolism. So there is a metabolism, there is formation of organic compounds, biomolecules which are in definite amount. It should not be less or more. It should follow a definite path for continuous reaction to take place, to be in a non-equilibrium state, so that the chemical reaction goes on continuously, and that leads to the living state.